So today we're going to spend some time again talking about meiosis and a little bit of how it's different from mitosis, which was chapter 12. And I think one of the most difficult parts about all of this is getting the vocabulary straight. So a chromosome versus sister chromatids versus homologous pair or homologous. Sometimes you just see it called now homologous chromosomes. Right, keeping all of that straight, I think, is one of the more difficult parts. In addition, the other vocabulary that we've already, or you probably have already spent some time with in chapter 12, that centromere and the kinetochore and the spindle fibers. Those are probably most of the new vocabulary words for these two chapters. So you need to remember that a chromosome is DNA plus proteins. The proteins are called histones, and these are proteins that help wind up the DNA and make it so that it fits inside a cell, inside the nucleus of the cell. Bacterial chromosomes do not have histones, so it's a thing that's unique to eukaryotes. So I'll just put that there as well. And remember, mitosis and meiosis are also unique to eukaryotes because they have linear individual chromosomes and they all need to be duplicated and then divided into 2 equal shares or 4 equal shares, depending on if we're talking about mitosis or meiosis. Now, the other thing that just flashed through my head while I was thinking about these vocabulary words are these 2 words, haploid and diploid. And in the most distilled sense, haploid means one set of chromosomes. I shouldn't say that. Let's say one copy of each chromosome that is in a population, right? So if I'm a fruit fly and I have 4 different chromosomes, haploid would be n equals 4, right? We also call this, you'll also see this referred to as just n. So n equals haploid. Now, diploid, a diploid cell or a diploid organism is going to be an organism or a cell that has 2 copies of each chromosome. I shouldn't say that is in a population. That's not right. We should say that is in an organism in a population. What I'm trying to get to there is, so again, if all fruit flies have 4 chromosomes, n equals 4. All fruit flies in a population, they all have 4 chromosomes, but each fruit fly itself is diploid, meaning they have 2 copies of each of those chromosomes. So that means they would be 2n equals 8, right? 2 copies of each chromosome that are in. So we do it like this. We say 2n is diploid. Now, individual cells are haploid and diploid at different times of their lives. And when you go to anatomy and physiology, they're going to talk to you a little bit more specifically about when the cells of meiosis in particular are haploid or diploid, because it's not as simple as it sounds. But we're in Bio 1, so we're going to do it the simple way, right? The idea is to just get you the survey course, to get you the basic mechanics so that when you get to the more advanced courses, you'll be able to sort it out with a little more detail. Sister chromatids. So let's go back, and I'm going to draw a little bit over here on the side. So I'm going to make a cell. Right? And I'm going to make a nucleus of that cell, and with a nucleolus, right? And now I'm going to change my color to, I don't know, purple, or it's not very bright. Let's do red. All right, so inside this nucleus, when the cell is just living its life before it is being triggered to either undergo mitosis or meiosis. So when this cell is just in G1, for example, it's a little thick, get skinnier. Why? Why can't it be thin? So my chromosome, I would have chromosomes like this. They're so thin and small. They're all wound up like this in the nucleus, and you can't see them as individual chromosomes, right? You just see them like as in prophase. You just see them in those like almost look like hair, right? But they're just getting ready to divide. And if I could look at just one of these chromosomes at a time, if I could look at just one of these chromosomes at a time, right? It would just look like this. It would be a single line or a single double-stranded piece of DNA. Let's say this is chromosome 1, right? So inside the nucleus in G1 before S, I would have 2 copies of chromosome 1. They're about the same size. They have their centromere in the same place, that little line that I've drawn to be the centromere. And it is not until S that they are going to each duplicate and become sister chromatids. So this is a cell, let's say n equals 4, just to be different. N equals 4. Let's say there are 4 different chromosomes inside this cell. So in its diploid state, it's just living its life and not undergoing mitosis or meiosis. 2n, or diploid, is going to be 8. And I just wanted to illustrate how we get to 8. So I'm going to make the chromosomes different colors to help with that a little bit, because they're basically different sizes, because we make our chromosomes based on the size and where we number them based on the size and where their centromere is. So this is chromosome 2, and I'm making it green. And I have 2 copies of chromosome 2 because I'm a diploid organism. And then chromosome 3, I'm going to make significantly shorter. And I have 2 of those. And chromosome 4, get purple, let's make this tiny one here like this. Right, so this is a cell where n equals 4. I have 4 different chromosomes. You can see them in different colors and or different sizes. And I already have 2 copies of each of them. 2n is 8. Now, it helps me, I don't know if it'll help you, but it helps me if I think about this in terms of my parents, right? If this is me, if this cell is one of my cells, this is me, right? And humans, of course, n equals 23, but that's way too many chromosomes for me to draw in order for this little demonstration, right? And it actually is 22 pairs of chromosomes plus a pair of sex chromosomes. So it's 22, we say autosomes, plus a pair of sex chromosomes. I'm just gonna write that. 23 different chromosomes total. So 2n, of course, would be 46. But again, that's too many to draw. So I just try to think about, this is, what if I was an onion? Onions actually only have 3, but fruit flies have 4. So what if I was a fruit fly? I would have 4 chromosomes. Actually, it's the same in fruit flies. 3 of them are autosomes, and there's 1 pair of sex chromosomes. And I have 1 copy of chromosome 1 that I received from my mother and 1 that I received from my father. That's what meiosis is all about. Meiosis is all about how did these chromosomes, or will these chromosomes get separated in me so that I only give half of my genome to my offspring? And again, it helps me to think about it just as me personally. And I think about, okay, in G1, before the cell goes through S and duplicates all these chromosomes, I already have one copy of chromosome 1 from my mom, one copy from my dad. If chromosome 1 happens to be the gene where the shape of my fingers is located, right? Depending on which characteristic my mother's or my father's is dominant, that's the one that's going to actually be expressed in my cells, in my organism. Okay, now, once S hits, once the cell is triggered to go into S, let's make that bright red so we see that, synthesis phase, every single one of these chromosomes gets duplicated. So my chromosome 1 now, I'm going to have 2 copies of chromosome 1, and that's when we see them like this. That's when books and when you actually can see it in the, that's when they're drawn like this, right? Or if we were drawing my stick chromosomes, I would literally just draw them like this. They're attached at the centromere like that, right? Usually we draw them as an X. Right? That's a duplicated chromosome, but I have to duplicate both of these. So let me try to just erase these chromosomes here. And oh, I got rid of all my words. That's a bummer. Right, so each one of these, I'm just going to make them X's. Each one of these would have to be duplicated like this. Now we call each of these pieces of the duplicated chromosome. That's when they're sister chromatids. Okay, each one of those. Together, together they're a homologous pair, or homologous chromosomes. And that's important because when we talk about, particularly about meiosis, about what happens in metaphase 1 versus metaphase 2, in metaphase 1, or such the anaphase 1, the homologous pairs are going to separate. In anaphase 2, the sister chromatids are going to separate. And I've only drawn one of the chromosomes. Remember, this has to happen for all 4 of the chromosomes that I have here. So I'm trying desperately to remember all of the things that I had over here on the left-hand side. So it's from, I think I had the word centromere and kinetochore on there as well. And I did want to just highlight with you guys the importance of those. So I'm going to draw just a separate chromosome here. I'm going to try to draw one that looks more like a duplicated chromosome. Yeah, not doing a bang up job, but get the general idea. So this area here, and it doesn't have to be in the center. It could be up here at the top, or way at the tippy top, or down here. That's what we call the centromere. Right, so write that out here. Now, the other important point here is where the spindle fibers attach is this, because it's protein, this collection of protein that forms this sort of connecting spot, or a C shape. I'm exaggerating it, of course. And that is going to be for that's the kinetochore. And that's going to be where the spindle fibers attach in order to pull the sister chromatids apart. Now, at anaphase 1 in meiosis, it's going to look a little different because we're going to have, just coming off of metaphase, we would have our chromosomes all lined up here on the metaphase plate like this, sideways. And they're lined up in their pairs, they're lined up in their tetrads because there's been crossing over that has occurred. So there's all 4 sister chromatids are here. So in this case, the kinetochore only forms out here and out here because anaphase 1, we're going to be separating homologous pairs of chromosomes. And then anaphase 2, we're going to actually, it's going to be set up like this, or more like mitosis, where we're going to separate individual chromatids. So I think that's all that I had over on the side there for a list of the words. Did I miss anything? I'm going to stop the share and ask, did I miss anything, any of the words that were over there on that side that I didn't describe particularly? I find it helps to grab some household objects and use them for the chromosomes and the sister chromatids. And I know it sounds very 7th grade, but I have some clothesline here that a long time ago, when I was a new faculty member, I dyed pink, and I have some straws that I cut up because I'm not going to use them to drink out of anymore, and some wooden sticks from the laboratory, right? And I actually pass these out when we have class, and I say, okay, here's your cell, n equals 3. I have 3 different chromosomes, pink one, a stick, and a straw. And this cell is 2n, it's a diploid cell. So I have 2 copies of each of these chromosomes, right? And then I provide you with another complete set, and I say, show me mitosis and show me meiosis. And all of the things that I'm showing you are chromosomes. So that way you can talk about S, chromosomes are duplicated, right? And if we're talking about mitosis, the chromosomes are duplicated, and during metaphase, they line up on the metaphase plate, and they're going to separate sister chromatid from sister chromatid, right? But if it's meiosis, they're going to form that tetrad, and crossing over can occur. Pieces are going to be broken off of one of these sister chromatids and swapped with another one. And then anaphase 1, homologous pairs separate, and then anaphase 2, sister chromatids separate. And we end up with 4 cells. Each one only has 1 copy of this chromosome because these cells are going to be sperm cells or egg cells, and they can only have 1 because they're going to meet up with another sperm or egg cell to go back to being 2n in the zygote, right? So at the end of meiosis, we end up with 4 haploid cells. At the end of mitosis, we end up with just 2 diploid cells. And I'm trying to just give a quick look here. If there's anything else that I was thinking about, particularly about mitosis. I think there's also a section in this chapter about karyotypes and what a karyotype looks like. A karyotype is just a picture, a photograph, or now they're mostly done on computers, of all of the chromosomes that an organism has, and they're metaphase chromosomes. So I think they're after the cell has duplicated all the chromosomes, and then they're squished out, and we can look at them. If a person has a gross genetic abnormality, meaning they are missing about 5 million base pairs, so about 5 million A's and T's and G's and C's, you can actually see them with a regular microscope, with a light microscope, that the chromosome is shorter or the banding pattern is different. Mutations or deviations, deletions, additions that are smaller than that 5 million, you can't really see them very much with a light microscope. What you can see are missing pieces of chromosomes, right? If one is significantly shorter, because there's a major deletion in the short arm of chromosome 13, for example, or clearly you can see if there's additional chromosomes or completely missing chromosomes. And for the additional chromosomes, we always use the example of Down syndrome, where a person is trisomy 21, which means they have 3 copies of chromosome 21. And also usually we mention aneuploidy in humans that come up in this chapter. No. so I won't. And then there's also a section about plants and fungus and other sexual life cycles besides humans and animals. So I wanted you to notice that there's mostly in this chapter, we're focusing on animals. And then there's also a discussion of alternation of generation with plants and some fungus. And I'd be happy to go over that with you if you want to go over. Okay, so in each different phase, they do all the same things, pretty much. Yes, Angelina. The cells, so there's diagrams in chapter 13, and I would spend some time studying those diagrams compared to the ones in chapter 12. What's different is how they organize. In meiosis, all 4 sister chromatids find each other during prophase 1. That does not happen in prophase of mitosis at all. In prophase of mitosis, the duplicated chromosomes are wherever in the cell, just floating around. In meiosis 1, in prophase, all 4 of these sister chromatids get together and form that tetrad, and that is when crossing over takes place. And that's a huge, huge important thing. When crossing over takes place, literally what happens is the sister chromatids, the arms of the sister chromatids get twisted around and they break and they exchange pieces. And they either are going to exchange pieces between their homologue, we say, between one of these 4, or very more rare, they exchange a piece with a neighbor from a different chromosome. Right? Most of the time it's within the homologue, and that's what we show in the textbook. That's why the chromosomes in your book in this chapter are blue and red. You can think about them again as being from your parents. Think about that cell being your cell. Like, okay, so I'm female, I'm going to produce eggs once a month, right? And so inside of my cells, the cells that are going to end up being eggs, that are going to end up being gametes, I have a copy of chromosome 1 from my mom, 1 from my dad. When that cell undergoes meiosis, I'm going to duplicate all those chromosomes. There's going to be crossing over in prophase 1. And this is how certain characteristics seem to skip a generation, especially with something particularly distinguishing. And you think, how did I get my grandfather's, my mother's father's nose, right? And that's how that happens. Right. Then these after prophase 1, they're going to line up, and metaphase 1, they're all still going to be together on the metaphase plate. Anaphase 1, they're going to separate, and now we have 2 cells that are technically still diploid. They still have 2 copies of each chromosome. And then there's going to be a little rest after telekinesis, after cytokinesis, a little bit of rest, telophase, cytokinesis. In mammals, in female mammals, that rest lasts from before you're born until until each egg or oocyte finishes its development to become a mature egg. So that rest period could be is at least like 10 to 12 years, or could be as long as 40 to 50 years, depending on when you hit menopause. Then each of these cells is going to continue on to meiosis 2, and the chromosomes aren't going to separate necessarily, but they're going to usually stay pretty close together. And then we're going to have metaphase 2, and then in anaphase 2, in each of these cells, the sister chromatids are going to separate. So we end up with 4 cells again, and each cell only has 1 copy of the pink chromosome, and 1 copy of the stick, and 1 copy of the straw. So that when they meet up with another human cell, that cell's going to have 1 stick and 1 pink chromosome. And now we're going to be a zygote. Now we're going to be a baby, right? We're going to grow into an embryo. All right, what did you ask me, Allison? 13.4. Let me take a look. Tiny, yes, you should. The importance of 13.4 is particularly that that is one of the major sources of variation. So I pretty much included that, I think, back in the general information, the beginning of the chapter, and when we talked about why is it important or what's the value of meiosis, that it produces those two genetically different cells for variations, the raw material for natural selection. So you're right. I did not expand upon it in the outline, but yes, you should read over that. It's almost like in a book, in a novel, when there's foreshadowing. That part of chapter 13 is to foreshadow what we're going to talk about in chapter 14, which is how genes are transmitted from one generation to another and how the variation gets transmitted. So we're going to talk about Mendel next. So it's kind of leading you into Mendel, because when we get to chapter 14, I'm going to draw pictures from meiosis again and say, remember those chromosomes and how they move? This is why it was important. So it's really important for as a source of genetic variability for diploid organisms. Okay, other questions about chapter 13? If not, I'm going to pause the recording and stop the recording, and then we can talk about any other questions that you guys have. All right.